Hello. We know that DNA replicates, and mechanistically it's a lot of trouble because we have a double-stranded DNA such as this. Although here it is depicted as two linear DNA molecules, it's actually coiled coil. It's a coil. That means one strand, each of them are passing over the other, and then yet it has to manage to give rise to two similar and identical molecules with the same number of bases, compositions, and also the sequence. And we know that it is happening through semi-conservative replication, which would mean that one of the strands is the parent. There are two strands in the parent. One is obtained by one of the daughter cell DNA molecules, and the second one is obtained by the second one. How does it perform all these things? Certainly, it's not just about polymerization. There should be many other factors that should allow this complex process of replication to happen. I am Dr. PCMR, Assistant Professor at Shastra. Now we will discuss about several enzymes. If not cover all of them, we will take into account some of the most important enzymes and then try to see their role and their probable, or in some detail at least. The first thing is the question of DNA molecule. That we have DNA and it is coiled coil in this way, and we should have it separated. This is a coiled coil like this, helical, and then we need to separate that. And for that to happen, there is an enzyme called as Helicase, which breaks the hydrogen bonds. Helicase is a hexameric protein which uses ATP to break through the hydrogen bonds. It will encircle one of the DNA strands and keeps moving in one direction, and because of which it will keep on interrupting the hydrogen bonds that are present between the two complementary strands, thereby separating the two DNA molecules. It is an active process because it consumes a lot of energy. Then say if Helicase was doing it moving this way in this direction, the supercoiling here, or the twists, are increasing, because if we have it rope and we try to pull, and the number of times it overlaps each other, they will become, the superhelical density would increase, and because of which at one point Helicase will fail to move ahead because the supercoiling is too high, superhelical density is too high. To relax that supercoiling, unless that supercoiling is relaxed, it cannot move further. Say here, assume this is the point of where Helicase is breaking and it's moving forward, and then the two DNA strands should be separated, because of which positive supercoils will accumulate. This is not only case during replication, but it also happens during transcription as well. So those supercoilings should be relaxed, which otherwise will not allow the denaturing of the DNA, the opening of the two strands for replication or transcription, as the case may be. So for that, there are enzymes called as topoisomerases, which will relax these positive supercoils to negative supercoils, or at least remove these positive supercoils in that location, which in a sense is actually, as a total DNA, if we look at it, is kind of inducing negative supercoils. So supercoiling, this topoisomerase 1 is a protein which will break. If there is supercoiling, the way it acts is something like this. It would make a nick in one of the strands and allow the strand that is here, this DNA molecule, to move, this DNA molecule or the strand to move downwards and twist, basically it would, if this is these are the two strands, there is a nick made by topoisomerase here, and this strand is allowed to move back like this and join here. So by that way, one twist has been reduced. That is how topoisomerase 1 works. There are also topoisomerases 2, which will actually make a double strand break. They would have broken this make a double strand break and then allow the other DNA to pass through and then rejoin them. That is an active process. Whereas topoisomerase 1 is supercoiling itself is a form of energy, potential energy. And all we need is to make a click, make a cut in case of topoisomerase 1 nick, and then this would be rotated and allowed to bind here, thereby relaxing the supercoiling. So topoisomerase, if we inhibit topoisomerases, the cell would have a lot of trouble making replication or transcription to happen. Then say we made the DNA and assume that Helicase has consumed a lot of energy and opened up the DNA. What would happen then? These are complementary strands. So the most likely thing that can happen, or the spontaneously what could happen, is that these two strands could come together again, and then renaturation could occur. And there is also one other risk, is that if we have single-stranded DNA too much exposed for too long, it is also a good likelihood that they might get damaged by endonucleases or any other kind of things. Somehow we need to have this, there should be protection, and most importantly, of course, is to prevent renaturation. Otherwise, all the work that Helicase has done is going to waste. So there are proteins called as single-strand binding proteins. They usually coat the single strand that is present here until these strands are polymerized. When they are polymerized, of course, single strands will no longer bind there. Right? So that is why single strand binding proteins are important. They form a kind of helix on the DNA, and they polymerase and they bind something like this. Now we have the important enzyme, that is the DNA polymerase itself. It is thought to be three-dimensionally structured. If you see, it is thought to be something like this, with the thumb region and then the finger regions and the palm region like this. And the DNA that is the active center is around this place approximately. As shown in the figure there, the DNA that is present here, and we have a template single strand on, that is the primer and template junction. So one of these will be, this is how the DNA template is present, and a new primer is growing in this active center here. DNA polymerase has to accomplish a lot more than just polymerization. It has to do it without any mistakes, and that is what is the important. That means it has to ensure that the base pairing is correct. Otherwise, the inheritance or the transfer of information would have gone wrong, or in other words, mutations would have accumulated. So it should ensure that the correct bases are added, and polymerization should occur, of course. And then there are a lot of dNTPs and also 10 times more of NTPs or rNTPs, which are subunits for RNA polymerization. So how does DNA polymerase choose only dNTPs and eliminate all the rNTPs? How does it check for the correct base pairing, whether the base pairing is correct or not? And by mistake, if a wrong base is incorporated, how does it remove the incorrect one? And not only that, it has to do very fast. It should be very fast, and it should have high processivity. We'll look into some of these in the following slides. First thing about polymerization. DNA polymerization, as we discussed earlier, through chemistry of the DNA synthesis itself. This is the 3' OH of the primer that is present, and here we have the incoming nucleotide. This is an incoming nucleotide with alpha, beta and gamma phosphates. So there are two metal ions, which you call it as metal ion A and metal ion B. They are important to for the catalysis of this polymerization itself. So the active site of DNA polymerase, something that is shown here, looks something like this, and here in this place we have the growing base pair, and this is the new this part is the new DNTP that is coming in. Here we have the template. This is the template, and here we have, this is the primer. So DNA polymerase somehow has to ensure that the T here it will be a perfect base pairing, and the important thing here is the alpha, beta and gamma phosphates are put in this something like this orientation, and we have the 3' OH here. So we have metal ion A and metal ion B. Metal ion A is important for activating, along with other amino acid functional groups of other amino acid residues in the of DNA polymerase, to activate this OH. And basically it would remove the proton and results in oxyanion. Metal ion B will stabilize these groups, the 3 phosphates, in such a orientation so that the oxyanion that has formed will form a phosphoester bond with the alpha phosphate, and which will also result in the cleavage of this bond here. And these two would be released as pyrophosphates, and the pyrophosphates would be acted upon by pyrophosphatase, which would be further broken down into 2 inorganic phosphates. So essentially this is how the polymerization would occur in DNA polymerase. The metal ion A and metal ion B mediate or activate the incoming nucleotide and also activate the 3' OH of the primer, resulting in polymerization. The next thing is, how can it correctly, DNA polymerase, how does it see that the correct base pairing is happening? The incoming nucleotide should be a T here. It should not be a G, C or A, which would have been wrong. So that is done basically in essence by the base pairing. If the base pairing is done properly, only then the 3' OH of the primer and the incoming nucleotide would have been oriented properly for the phosphodiester bond formation to occur. If the base pairing is wrong or it is incorrect, it's not happening, it is not the classical Watson-Crick base pairing, then the incoming nucleotide would not have been in this proper orientation, because of which the phosphodiester backbone, the bond would not have formed. That is how DNA polymerase to a large extent eliminates anything wrong incoming nucleotide. I mean, it's a random process. Basically, in the active site, any of the 4 nucleotides could fit in, and the DNA polymerase would wait until the correct one comes, and this is actually a very rapid process. Although there is a certain degree of accuracy, there are at times mistakes that do happen, because DNA polymerization is happening at a very high rate, 10,000 bases per second or so. The second one is, we discussed that there are DNTPs and there are RNTPs, ribonucleotide triphosphates. So if there are the RNTPs actually are in, we should remember that especially if it is in eukaryotes, there is a nucleus, and transcription and transcription and replication are all happening in the nucleus itself. So there is RNTPs, the transcription is happening many more times than replication. In a cell cycle, replication would happen once, but transcription continuously happens, although it's not complete genome, but majority of the genes are expressed. So there are about 10 times more RNTPs compared to that of DNTPs. And how does DNA polymerase? The difference between RNTP and DNTPs is just that one oxygen in the 2' carbon of the ribose. How does it discriminate? How does DNA polymerase discriminate DNTPs from RNTPs is the question here. In this case, the DNA polymerase has certain residues here, which is designated as discriminator region. And if there was OH present here, it would not fit into the active site properly, because of which again we would not have any phosphodiester bond formation in this case. And that is how the RNTPs are eliminated, and only DNTPs get polymerized into the DNA, into the primer or the growing primer. The next one, as I said before, although there are a lot of mechanisms to proofread or to ensure that only the correct bases are coming, there is always a problem that there is a possibility of a wrong base to be added. And then what does DNA polymerase do? As I said, the speed is very high. So if we had something like this, say we will cook up this and CATAG. Now DNA polymerase has to add, and DNA polymerase would add C, T, and if it added something like cytosine here, which is a mismatch, this is a wrong. This is wrong and should be corrected. DNA polymerase also has an exonuclease activity. If we again visualize DNA polymerase to be this way, around this place we have the polymerization active center. Around this place we have something like the exonuclease activity. As the name indicates, exo means out and nuclease activity is breaking it or cutting the nucleotide. So what DNA polymerase does is, if DNA polymerase was moving this way and it is wrong, it would move back and eliminate this region, that one base that has been added. And again DNA polymerase will start adding the bases correctly. This way DNA polymerase is a self-correcting enzyme, and it's something like it has its own introspection to track its own mistakes and be able to do that. How can somebody move forward when you're also moving backwards? You're moving backwards when we are trying to correct our own mistakes. And DNA polymerase it doesn't move back all the time. It only moves back when it does a mistake, and that is important to move back, because mistakes should be corrected. Otherwise we are making a wrong product or a bad product, which will make the whole efforts of replication futile. So when DNA polymerization is happening here, it is fine. When the DNA that is growing makes interactions with the palm region, thumb region, and also fingers. There are many more aspects that we are discussing. So if there is a mismatch or a wrongly incorporated base, then the interactions are reduced between the new DNA that is being formed with that of the DNA polymerase, the palm region, because of which it could slide to the region of exonuclease activity. And during that time, if during the exonuclease activity the wrong base is eliminated, because of which it would regain its activity or affinity for the palm region or the active site center, polymerization active site center, and then polymerization will resume. So majority of the time DNA is here, but every time or majority of the time when there is mistake, it comes back. Somehow it will get exonuclease, the wrongly incorporated base is removed, and then it goes back. And that is how DNA polymerase is a good example of how or why introspection is important. There is one more aspect is DNA polymerase is the DNA should not move out of the DNA, or DNA polymerase should be able to add as many as possible, because it is the genome is huge and we have to add a large number of bases to the DNA during replication. In that case, the DNA should be bound with the enzyme, and processivity is a term used. It is about the number of bases added or number of reaction cycles performed before added before detaching from the DNA molecule here. Number of bases added before detaching from DNA. Okay, so if there is a DNA molecule and there is an enzyme, it adds about 10 bases and dissociates, compared to another enzyme, say enzyme B, which adds about 100 bases before it detaches. Then you can say that B has high processivity compared to A. For DNA polymerases, then they are adding thousands of bases per second, and they have to add a lot of DNA before they dissociate, because to find the place and again resume is an expensive endeavor. So it would be nicer or better for DNA polymerase to bind to it and keep on polymerizing without detaching. For that, there is an enzyme that is called as sliding clamp, and the sliding clamp binds to, encircles the DNA like this, something like this, and to which DNA polymerase is bound. So now the probability of DNA slipping out is very less. Which means there is increased probability of DNA polymerase bound to the DNA, and then polymerization would occur. The DNA keeps on sliding on the clamp keeps on sliding on the DNA polymerase. So that is how the DNA polymerase or sliding clamp increases the processivity dramatically of the DNA polymerase. In prokaryotes, we just call it as sliding clamp. In eukaryotes, we could say it is labeled as pCNA, proliferating cell nuclear antigen, which would mean that it is present in the nucleus. It is present in proliferating cells that are rapidly dividing, which would, if they are dividing, replication must be happening, and during replication you need sliding clamp. And in eukaryotes, if you're finding people find this pCNA, and that's why they call it as antigen maybe, and it is found in rapidly replicating cells, and that is pCNA in eukaryotes. There are, although we discussed DNA polymerase, there are many other types of DNA polymerase. It's not one type. In eukaryotes and prokaryotes, there are multiple types of DNA polymerases. Say in E. coli or in bacteria in E. coli, you can find polymerase 1, 2, and 3. They're Pol A, Pol B, and Pol C. Those are the names of the genes. Number of subunits is also given here. What we need to remember is they all have the polymerization activity, of course. That is why we call them as DNA polymerases. How do they differ is there is also we discussed about exonuclease activity, and we said something like this. This is the 3' end, and DNA nucleotides are being added in this direction. And if a exonuclease activity was performed and one base was removed, that means DNA is moving from 3' to 5', and we then call it as 3' to 5' exonuclease activity. That is, majority of the DNA polymerases have 3' to 5' exonuclease activity, which is also referred to as proofreading exonuclease activity. There are some DNA polymerases which also have 5' to 3' exonuclease activity. That is by DNA polymerase 1. So DNA polymerase 1 is something that will excuse me. So DNA polymerase 1 is special because it has polymerization activity, and it's better to remember 5' to 3' polymerization activity. It has exonuclease activity, proofreading exonuclease activity. It also has 5' to 3' exonuclease activity, which will become handy for us in some time. And there is something else we need to look at, the speed of polymerization. DNA polymerase 1 is little slow, or the slowest one. At the number of bases it can add is about 20 bases per second, whereas DNA polymerase 2 is a little faster. But look at DNA polymerase 3, because it can add between somewhere 250 to 1000 bases per second, and that is the reason why DNA polymerase 3 is important for replication. The other two, DNA polymerase 1 and 2, are specialized for doing small jobs, cleanup jobs, something like the repair, DNA repair. If there is any damage, gaps, then they are good to fill in those gaps there. And the other point is the processivity. You can compare the processivity of these three enzymes. You see, this 500,000, DNA polymerase 3 is about 500,000 or above that. It's mostly because of the sliding clamp, its association or ability to bind to the sliding clamp. Whereas the polymerase 1 and 2, they are only for small patches to be filled in or repaired. That is where they are involved, so they don't need to have very high speed or very high processivity. There are also other polymerases, which are polymerase 4 and polymerase 5, which are also involved in DNA repair. It's called as TLS polymerases, that is translation. Translation synthesis. Lesion means injury, or if there is a DNA, and there is this, and here is in the template there is a problem. We will discuss this when we are discussing about repair, but for the moment assume that there is a damage here. Damage in the sense the nitrogen bases for some reason are unable to form the bind. So if we take any of the DNA polymerases 1, 2 or 3, they have exonuclease activity. They don't want to do any bad job. If they do, they want to do a good job and perfect job. And if there is a DNA timer, for example, thymidine dimer, because of which the DNA polymerases, the normal ones, cannot function here because they are too perfect for this scrap job. They cannot add anything just like that. That is where the DNA polymerases 4 and 5, which are involved in repair, they don't care about it. They just add something here and fall off. So this becomes important in the sense that there is always is something better than nothing, or nothing is better than nonsense, right? It is a matter or a compromise between speed and accuracy. Sometimes we need, sometimes speed is preferred, most of the times accuracy is essential. So if we take these enzymes 1, 2 and 3, say 1 and 2 are not so fast, but they are highly accurate. If you take 3, it is very fast and it's moderately or relative to DNA polymerase 1 and so on, it is accurate enough, at least if it is not the most accurate one. What is the use of an enzyme that doesn't have speed or doesn't have accuracy? That's an important question, right? And that is the use that comes here. When there is severe damage and there is no way but to fill up something, that is where these enzymes, the DNA polymerase, the translation DNA polymerases, are very useful. They just add something, wrong basis or whatever basis, and their process speed is less, processivity is very less, and they fall off. And from there on, the DNA polymerases could take up and will keep on synthesizing. I hope you enjoyed this. Thank you very much. We'll see you again later.